this section I will take up the tools which we have at Ncbi, and these are the several tools available at Ncbi, and I just listed them, all of them here. We don't intend to do all of them, but if you notice that BLAST is the most common one here, and in several of the tools BLAST is again and again used. So what we'll do is we'll talk about BLAST in this section. So what is BLAST? BLAST is, full form is Basic Local Alignment Search Tool. So BLAST is very popular, I said, and it is one of the most widely used bioinformatics program for sequence searching. So what is BLAST doing? BLAST creates local alignments. So what are local alignments? Local alignments are looks for similarities between two sequences that may not be best or optimal. So alignment can be optimal alignment or it can be local alignment. The local alignment does not think about the full sequence alignment. It will find the places where it is matching and align. But in the global alignment, we look for best or the optimal alignment. So BLAST creates local alignments. And why BLAST gained so much of popularity and is used very extensively? Because the sequence searching before the BLAST algorithm came was done by Smith-Waterman algorithm. It's not that Smith-Waterman algorithm was lacking anything, but the thing was that this Smith-Waterman algorithm was very time consuming, and it was looking for accurate, absolutely accurate matching of the sequences. So when it was looking for accurate matching, it was taking time. So BLAST came in 1990, and it was designed, it was a paper published by Stephen Ashul and David J. Lipman, and there were few other members in their group. This paper came out in 1990, and it was such a revolutionary research that in Journal of Molecular Biology, when it was published, it was cited over 75,000 times. So what did BLAST do? BLAST made the searches faster, and the drawback here was it was not guaranteeing the best or the optimal alignment and the query sequence, but it was fast, and it did not ensure the best performance, but it was fast and it saved a lot of computer time and power. So if we So what does BLAST do? What do we do in BLAST? Is BLAST compares the sequences. BLAST takes a query sequence, compares it with millions of sequences in the GenBank database, and by constructing local alignments, it lists those that appear to be similar to the query sequence. This is called as hit list, and it tells you why it thinks that they are homologous. And the point which is to be noted here is BLAST makes suggestions, and you have to make the conclusions. You have to decide whether it fits, the suggestions of the BLAST fits in your criteria or not. So how BLAST does this thing is that when the two sequences in sequence searching in BLAST we are trying to match, what it does, it quickly finds where it is matching few words, and that we call as word. After it has found one word, it would find the pair of the words of the same length, and after it has found word pairs, it will calculate a score for this word pair. So the score, for calculating the scores, like we take the alignment of these two words VALMR, VAKNS, So it will calculate a score and it will use a matrix, similarity matrix, PAM matrix 120, say for example here, and on the basis of high scoring sequence pairs it will decide the hits, meaning the high scoring sequence pairs are called as hit. So once it has achieved the hits, it will tell you the sequences which are similar to this, to your searched sequence. So why should we use BLAST is suppose I am working on some gene or maybe some, like I take the example. So why should we use BLAST? First is for finding the model organisms for study of a disease. Say for example, we are talking of cystic fibrosis is a genetic disorder and that affects human also, right? And my question which I am trying to ask is, can yeast be used as a model organism to study cystic fibrosis? And if yeast contains a protein that is related or maybe homologous to the protein involved in cystic fibrosis, then yeast can be used as model organism to study this disease. And when we study this protein in yeast, we can know about the function of the proteins in humans. So if we are searching for a model organism for study of a disease, BLAST will help you. Then also BLAST helps you when you are trying to find the homologous proteins and genes, meaning with finding the homologous genes we would know where is the common ancestor, meaning how and where the sequence diverged. So we will be able to decipher the evolutionary relationships among the sequences. So we can know the sequences, the regions with the similar structures in the sequences, the regions with the similar functions. So this homologous information about the homologous genes and proteins also can be retrieved from BLAST searches. So in general, when we are doing a BLAST search, it is better to compare proteins than genes because of the obvious reason we have more variations in amino acids than in the nucleotides. So it is advised to do it with proteins. Then the criteria which we use for considering two sequences to be homologous is that if it is a case of a protein, if the amino acid sequences are at least 25% identical, we say that protein is homologous. If the DNA sequences are at least 70% identical, we would say that the DNA sequences are homologous. And we should at least try running a 100 amino acid long sequence in the BLAST. That's a web view of BLAST. And the point which I thought we must note here is that the bioinformatics research is the latest one. Every day it changes, like the information in Google. Like here, if you see that the BLAST has integrated the data of RefSeq, so that you not only search the GenBank sequences which are owned by someone, it also searches the RefSeq sequences in the database. Then we have several types of BLASTs which we can use, and we can choose the flavor of BLAST which we want to use. Like we have online version available. In the online version also we have several options, like we can do nucleotide BLAST, meaning we are searching the nucleotide sequences. We can do nucleotide, translate the nucleotide and nucleotide to maybe protein, or maybe we can use protein BLAST. So, and we can also do the genome BLAST, and we can use the sequences of different microorganisms, or maybe mouse, rat, humans and several microbes. So it all depends what you are trying to isolate or search. You can use any of these versions. We also have several standalone and API BLAST, like you can download the BLAST on your laptop, or maybe you can use the cloud version, or you can use API as it is. There are several, the Another flavor of BLAST is that there are specialized searches possible, and they are used for specific purposes. Like of these, if we take some, like the say for example CD search, it takes the conserved regions. So apart from BLAST, it will tell you the conserved region. It will, if you are doing vector search, vector BLAST, what it will do is it will tell you the vector contamination. So we have different types of BLAST, and they can be of specific purpose. So these are the specialized BLAST searches available. BLAST is improving itself every day, as I said earlier, and this is a web view of BLAST where you can enter your sequences. But the important thing to be noted here is that BLAST is consistently working on reducing the time taken in the search, because the data is increasing and we need to work on so it has designed special default parameters. Doing that, you can reduce the search time, and it also tells you several options with which you can filter and reduce your time of search. So yeah, so this is a simple search which I thought I'll show you. Otherwise I am planning to run a small MP4 which I saved for the run which we did. So we can enter either the accession number or the sequence directly in the search box, and we usually select the NR, that is the non-redundant data available, and we take rest of the values as default mostly. And we are doing mega BLAST because it looks for the sequences which are more than 95% similar. So what we are doing in this search here is that we'll be searching the sequences which I had submitted earlier, so those sequences are the more the conserved sequences which are used for molecular identification purpose. So for there, it would be appropriate to use mega BLAST. So when you submit, when you press the submit button, you get the output. So I think I'll do it again with the help of a video. So once the video is played, we'll do all the steps again. So in the BLAST output, then it will tell you how long are the aligned segments, and did the BLAST introduce the gaps in order to align, or how similar are the aligned segments. So the output which you get in BLAST, I'll just show you this in the video, how you get the output and how do we proceed. So in this MP4 which I had recorded, first I tried to find the nucleotide, meaning I thought we first isolate our sequences at the nucleotide database. So what you have to do is, you don't have to do anything, is you just select nucleotide at the top, and you can write the accession number of our sequences, and that will take you straight to our sequences. So it takes you to our sequences, and this is our sequence which is opened up. You can have a look at this. You get the complete sequence, and you can download it from here for making a BLAST search. So you get all the features of this sequence, and you can use this for I thought if we use this for BLAST, it will be nice. So I was working on the molecular phylogeny. I was trying to identify my spores of AM fungi. These sequences we have isolated from AM fungal spores, and they refer to the conserved region. So in this section of BLAST, you would know how to search what happens when we search conserved sequences. So apart from this, in this you can gather a lot of other information also, like you can do BLAST here itself, or maybe you can know the conserved domains of your sequences in this one also, but we will not do it this way. We'll go back and we'll search the BLAST interface, and I will show you, this is the home page of the BLAST, and I just showed you this earlier. So, and this can show you different flavors of the BLAST. You can have a look at them here. So we have different flavors of the BLAST and we can choose any one of these, but for our work where we are going to search the sequence, nucleotide sequence, and that's an evolutionary sequence, we will use the nucleotide BLAST. So how we have to do it here? We will enter the accession number at the search box and we will press the BLAST after selecting the database and selecting the mega search, because the sequences are expected to be very similar. So it took some time to search and it gave you the output. The output, you can see that it found a lot of sequences and these sequences are quite similar, and if you look at this, the output is in several form. You saw a lot of sequences and the top ones are 100% similar. So it suggests that the similar sequences from the same spores were already deposited at GenBank. And if you can read out, the entire sequence was matched. So it gives you results in graphic form also. It gives you result in the form of this sequence. In the graphic form, you can, with the help of the color, it gives you different colors based on the number of hits which we have. So you get most of the red color here. Why? Because we had a lot of sequences which were similar. You can also know the taxonomy of the organism here itself in the output, and this shows you the alignment, that you can see the exact alignment where the sequences had the gaps or where the sequences were exactly matching. And you can also have a look at the taxonomy. You can also save your searches here and maybe for next time to work, and you can save your searches. So you can save your searches, and I will just quickly summarize about the results. So you can save your searches, and in the BLAST output we get what? We get a hit list. So it gives you the best matches, which we call as hit, and this hit list provides you an accession number of the sequences which are hit or maybe similar to the sequence of your search. It gives you a description of that sequence. It gives you a e-value. What is e-value? E-value is an indicator of how good a match of the query sequence it is. This e-value is usually, meaning it should be 0 if a sequence is similar. Then it also gives you a score. A score meaning score for the link to this alignment, meaning what score it gave for this search. And if we like to know a bit more about the e-value, e-value refers to the chance that the match is not random. So the lower the e-value, better it is. E-value 10 to the power minus 4 is considered to be cut-off point, meaning we do not consider any e-value which is less than 10 to the power minus 4. And e-value of 0 means that the two sequences are statistically identical. Mostly people use e-value as a first indicator of similarity. The alignment, if we look for the alignment, we look for long regions of the alignment, the regions where we have fewer gaps, and the percent identity I told you already. For proteins it should be greater than 25%, and for DNA it should be greater than 70%. So to conclude or to summarize or to get the gist of this, what we understand is BLAST can make you suggestions. BLAST can make you suggestions, but you have to draw the conclusion based on your interests and your study area, your objectives of the search. And we have to look at e-value, we have to look at the graphical display, and if necessary we can have a detailed look at the alignment in the BLAST. And then finally you can make your best guesses. So that was about BLAST, and with this one we have come to an end of this presentation. And to summarize once again, we learnt about some of the databases of Ncbi, and we surfed through an important tool at Ncbi, that is BLAST. I hope you enjoyed this lecture and could understand everything whatever have been told, but still if you have any questions, I would be very happy to answer them. Thank you very much.